Good morning, everyone. Oh, come on. We have breakfast for you. Good morning, everyone. There we go. I'm very honored and happy to be introducing my role model and mentor, Dr. Elizabeth Stoner, or Betsy. So I'm just going to be just a little quick overview because she has done so many things. So Dr. Elizabeth Stoner is an assistant professor in the Department of Natural and Applied Sciences at Bentley University in Massachusetts. Her research focused on the ecological roles of benthic invertebrates in coastal marine ecosystems. Specifically, her dissertation work was on Cassiopea and their potential as bioindicators of nutrient enrichment in Abaco Island in the Bahamas. Sorry, being old is hard. So some of her current work evaluates the role that ecological memory plays in seagrass beds and how interacting stressors may affect that. Much of Dr. Stoner's research has examined the effects of human-induced nutrient enrichment on the abundance and ecological impact of benthic jellyfish. She has found that elevated nutrient levels correlated with increased jellyfish densities and sizes, which in turn are affected in the composition and diversity of the benthic fauna in these habitats. She has also contributed to studies on mutualistic relationships between jellyfish and symbiotic algae, investigating how these interactions influence nutrient cycling and energy. In addition to that, she has been involved in teaching environmental science and oceanography courses at Bentley University, where she teaches primarily business students. She emphasizes the importance of understanding human impacts on marine environments and explores ways to mitigate these effects through informed business practices. Ultimately, her work highlights the interconnectedness of human activities and marine ecological processes, and stressing the need for an interdisciplinary approach to environmental conservation and sustainability in the nearshore marine environments. She's a great mentor, a wonderful ecologist, and I hope you enjoy her plenary talk. Oh, and she inspired my dissertation.
Well, now Natalia continues to inspire me. Thank you for that. Good morning.
Good morning.
I'm really excited to be here. I have been working with Cassiopea for almost 2 decades, which talk about feeling old. And this is the first time that I have been at the Cassiopea workshop, which I was telling Aki, I don't actually know how that's possible, but I'm pretty sure it's now my new favorite conference based on just the incredible conversations I've had already with so many of you. Sheila, we're already talking about establishing a new Cassiopea, what do we call it? International Institute. The Cassiopea Institute. So it's just been so much fun already. And I also hear that in years past, shoes were optional. So I'm pretty sure that that absolutely officially makes it my favorite conference. All right, so today I'm going to speak about Cassiopea ecology. That's what I've based my entire career on. And so we're going to really get into the weeds on Cassiopea blooms. Are they actually happening? Are they being driven by anthropogenic activities? What is the role that Cassiopea and potentially their bloom events play in nearshore marine ecosystems? And I'll talk about some of our latest work looking at whether or not there are top-down consumptive effect-based controls on Cassiopea. So getting into it, as I said, I've been doing this work for, can I change it? Oh, maybe just I do that. Yeah, okay. I've been doing this work for quite some time. And when I started getting interested in Cassiopea ecology in the early 2000s, there was a lot of information that was popping up in the literature and also in the media about jellies and their blooms and jellyfish are taking over the world. Does anyone remember this? Right? Okay, show of hands. Who remembers all the chat about jellies taking over the world? And it's funny because it's still happening. There's still all of this media frenzy on gelatinous zooplankton blooms. All right, so this is, I pulled just some screenshots from the media when I Googled this several weeks ago when I was putting together this talk. This came up. This says, Resorts on Spain's Costa Brava struggle with invasion of jellyfish as seas warm. This one, closer to home for me, jellyfish on Cape Cod sting hospitalized teacher. Her response is science. Jellyfish, if you can't beat them, eat them, okay? Jellyfish are showing up in unexpected waters. What's going on? That sort of hype of, oh, they're taking over. What's happening? My personal favorite are heartless imbeciles really taking over the world. I thought this was just exceptional, just exceptional. All right. So again, really driving home the point here that there's been a lot of discussion about whether or not jellies are blooming, and a lot of discussion on whether or not they're blooming as a function of anthropogenic activities. All right, so this paper came out by Kylie Pitt and others in 2018. And they're talking about this frenzy on jelly blooms. And this figure just shows on the y-axis number of papers put out, years on the bottom. So you can see that more and more literature was coming out on jelly blooms really when I was starting my career in the early 2000s. The black bars indicate the number of citations that report that jellies are blooming because of human activities. So you can see as time goes on, more than half of those citations are indicating that jellies are in fact blooming as a function of anthropogenic activities, all right? There's a lot that that this paper talks about. One of the big things that big issues that they raise is whether or not this is actually happening, okay? So a lot of the work has been largely correlative. And I really got interested in trying to ascertain whether or not jellies are in fact blooming, so increasing reproductive output and increasing population densities as a function of anthropogenic disturbances. And around this same time that I started getting really interested in these questions, this paper by Anthony Richardson and others came out in Tree called the Jellyfish Joyride. Does anyone remember this paper? This was, yeah, show of hands. Okay, this was a big one. This one really, I think, got a lot of attention, in part because what they were saying is that jellies thrive, and I want to be clear here, pelagic jellies. A lot of this literature is focused on pelagic jellies. What they were saying is pelagic jellies thrive in these human-dominated systems, all right, and that they're a weedy species. They say this, they're a weedy species that may be globally increasing. And what they ultimately claim in this paper is that jellyfish have set us on this joyride. So as human activities, for instance, overfishing, reduce predators of pelagic jellies, that in turn allows jellies to proliferate, and then they drive this positive feedback loop, ultimately creating a system where jellies will outcompete other organisms such as fishes, and ultimately continue in this jelly-dominated system. And this is a really great, actually more recent infographic demonstrating this concept that through anthropogenic activities, which we'll get into in a minute here, but which you can see below, through these anthropogenic activities, you have this shifting baseline where you go from more complex marine food webs down to really simple gelatinous-based food webs. Jellies being villainized here. I don't know how I feel about it. All right, so what are jellyfish blooms? I am sure, in fact, I am confident that everyone here knows what a jelly bloom is, but just as a refresher, there are two types of jelly blooms. There are apparent and true blooms. So apparent blooms can be where there seems to be a higher abundance of jellies within a given area, and these are typically driven by wind, currents, perhaps the jellies are moving themselves, all right? True blooms, which is what we're really, really interested in, is what I've been really interested in, I should say, is jellies increasing in terms of their overall densities because they're they have increased reproductive output, okay? And again, there's been a lot of discussion about, is this happening? Is this not happening? We still have no consensus globally on whether or not jelly populations are increasing. There was a paper that came out by Rob Condon, 2013, and others that indicated there is a slight but significant increase in global jellyfish populations. All right. They did also caveat this and said that jellies do seem to undergo a periodicity of about 20 years in terms of their bloom events. Okay. And that this is really context dependent, right? This is really dependent on the specific species, on the location where that species is located. So we don't really know is the answer, okay? We don't really know if jellies are increasing globally, but we do know that there are many jellies that do seem to be increasing in nearshore coastal marine ecosystem environments. And so that is what I've spent a lot of my my career getting into with Cassiopea. One non-benthic jelly example right now is the clinging jellyfish. This is an invasive jelly. There's a lot of work on it right now in New Jersey, closer to where I am. And they do seem to be blooming. Possibly, though, this is still correlative as a function of excess nutrients. All right, so again, you probably all know this very, very well, but some of the more common anthropogenic drivers of jellyfish blooms that have been discussed, these occur in isolation, but also occur often together and can have potentially synergistic effects on marine ecosystems. So things like overfishing, right? Taking away competitors of jellies, but also predators of jellies. Ocean warming, so we know from a lot of the literature that jellies like it warm, particularly in their asexual reproductive stage where they'll strobylate more in warmer water. Habitat modification, also called ocean sprawl. So a lot of jellies, particularly the polyps, will grow preferentially on artificial substrate. We find it more on dock pilings, where a lot of the spots where I do my research in the Bahamas and South Florida, we find it a lot on boats, docks, and then nutrient loading, which is what we'll dig into a little bit more today. And Natalia is going to talk to you more about it in her talk later. And there have been a number of papers exploring the ecological consequences of jelly blooms. Again, really want to emphasize here that primarily a lot of this literature does come from pelagic jellies, okay? So our Cassiopea have received a lot less love when it comes to understanding their ecology. But what we do know is that there are papers that indicate that there's increased competition for food. There's direct mortality, of course, by predating on fish. So jellies will eat more fish. Competitions for space, specifically the polyps. All right, they're competing for space with other sessile organisms. Shifts in nutrient dynamics and dissolved oxygen. So, I mean, we know from the Cassiopea literature that increased Cassiopea densities will have increased oxygen production in sediments and the sediment water interface. All right, but that's also been somewhat documented in pelagic jelly literature. And then also effects on foundation species and community dynamics. I won't be talking about that much today. This is a lot of what I've done my research on, is looking at specifically how Cassiopea may affect seagrass community and ecosystem dynamics. All right, this is actually from a paper that we put out in 2014, where we experimentally manipulated Cassiopea densities as well as nutrient concentrations to see if that would influence various aspects of seagrass communities and seagrass productivity as well. I'm sure a lot of you have seen Cassiopea in seagrass beds, so you know that their presence can potentially influence seagrasses. What we ended up finding, one of the things I wanted to highlight, is that we were looking at grazing intensity on seagrasses. Grazing is a really important control in seagrass beds. And we found that in plots where we just had increased jellyfish abundance, okay, so we artificially increased jellies, compared to plots where we had just elevated nutrient concentrations, and where we did both, we elevated jellyfish and nutrient concentrations, we saw that grazing intensity was significantly lower, over 90% lower in our jellyfish plots, okay? And we see this even anecdotally, where fishes will avoid swimming through really dense thickets of Cassiopea. They'll swim away. I've actually seen little yellowfin mohara swim through Cassiopea when I've inadvertently disturbed them. So a lot of mucoid exudate, and they were hit with the mucus, got stung, and died. So really interesting stuff. And I'm getting ahead of myself because I'm already talking about Cassiopea because I love them. But again, in general, this is what we know from the pelagic literature. All right, and last but not least, we know that true jelly blooms can also have economic consequences. So in, I believe this was 2023 and 2024 in Norway and Scotland, over 3 million salmon were killed from a jelly outbreak there. They were not recoverable. This is, apologies for the somewhat uncomfortable picture, but these are salmon that are just completely broken apart from those jellies. I forget which species got them. I want to say it's mauve stinger, but I don't think that's true. All right. And then in Korea, jellyfish damage to fisheries comes in at between 2.1 and 25% of the annual production value of those fisheries. So millions of dollars lost annually related to jelly blooms. And then, of course, there are also issues related to desalination plants, power plant closures from sucking up jellies in intake valves for cooling, and then tourism concerns. Especially, this is a real concern in small island developing states. Natalia mentioned, I do a lot of my work in the Bahamas. I've talked quite a bit with Bahamian government officials about concerns related to really dense populations of Cassiopea in heavily tourist-based areas. They have asked me if removal programs are possible. So this is absolutely a concern for a lot of folks. Okay, so just to recap so far, where are we on this jellyfish joyride? There is this perception that they're increasing globally, largely attributed to anthropogenic activities. Global jelly populations do oscillate, right? Again, they do undergo this periodicity, though we do know that coastal increases are occurring. And a lot of what Natalia is going to talk to you about today is about that. So ultimately, more work is needed on this. And this is where finally, we get to Cassiopea, okay? The star of the show. So again, there's this paucity of literature on these amazing animals and their ecology and whether or not these animals are in fact blooming. They are global, I don't need to say any of this to you, but they are globally distributed in subtropical and tropical environments. A lot of the work that I've done is in seagrass beds, but of course we find them in mangrove ecosystems, corals, they're everywhere, bare bottom, bare sediment. Some are invasive. I think we have a talk later today about them as invasives in Hawaii, which I'm really excited. Is that you? I'm really excited about that. And then they do, of course, a lot of you study their symbionts. So cool, such an interesting system. And those symbionts, I've done some work looking at how they may influence Cassiopea bloom dynamics. So how much do we know about Cassiopea blooms? When I started working on these guys in the 2000s, we really didn't know much. Richardson, in that Jellyfish Joyride paper, did sort of put out a call to action, if you will, and said there needs to be more fundamental research on jellyfish ecology, their complex life cycles, and their roles in ecosystems. All right, so I really set out to do a lot of that. And I've been very fortunate to work with some incredible people like Natalia on trying to better understand this. One of the things that has become very clear through our work, and this is from Natalia's paper that she put out last year, is that there's not a lot on them. There's not a lot on their ecology. All right, so this was, she looked at close to 200 papers and found that roughly 10 have focused on their ecology and 1 has talked about their blooms. Was that mine, Natalia? Yeah, okay. So, so again, what is happening here, all right? And if they are blooming, how might that impact their role that they play in these nearshore ecosystems, in particular in nearshore food webs? I've been really interested in that. So generally speaking, my research on Cassiopea has 3 main tenets. The first is, are they blooming? And are they blooming because of human activities? The second, which I sadly won't have time to talk about today, but I will talk your ears off about this if you ask me. Do they have potential effects, maybe deleterious effects on nearshore ecosystems? And third, what I'm going to talk about today a bit, some of our latest work looking at the role that they play in marine food webs. Are there top-down controls on these organisms, right? I get that question a lot. Well, if they're blooming, is there any way to control them, right? Are there controls on these blooms? Okay, so the first part of what I'm going to talk about today is some of the work that we've done looking at these blooms. All right, so first I'm going to talk about some of our work that was done in 2011 on Cassiopea related to human population densities. And then the second part is looking at whether or not anthropogenic nutrients can be a driver of Cassiopea growth. So this work, as I said, we put out in 2011. And this was really getting into, okay, are Cassiopea found where there are people? Okay, so are they more abundant and are they larger in areas that have more human disturbance? So if we look in the figures up top, on the left-hand side here, sorry, apologies for the small text, but it says Cassiopea species density and high human population density and low human population density on the x-axis. We sampled in the Bahamas in 10 different sites using human population density as a proxy for disturbance, and ultimately found that Cassiopea were 95% more abundant in these human impacted sites compared to low human population density sites. Similarly for size, okay, so this is size frequency distribution over here on the right, looking at Cassiopea biomass. And similarly, we found that Cassiopea were 95% larger in these human impacted sites. Again, I want to emphasize this is correlative, right? We weren't looking at whether or not Cassiopea were actually increasing in terms of the reproductive output, but this was a really good start, we felt, to try and establish whether or not there's this relationship between Cassiopea and us. We also sampled for nutrients in water and submerged aquatic vegetation and found that Cassiopea density was positively correlated to total phosphorus concentrations in the water. Interestingly, we know that the zooxanthellae do preferentially take up ammonium as a form of nitrogen. And we found that sites that had these high ammonium concentrations actually had consistently low densities of Cassiopea. What we're thinking there is that possibly could be indicative of locally depressed ammonium from zooxanthellae use. but weren't really able to get into the weeds too much more on that. Until a few years later, I was really interested in this relationship with nutrients and Cassiopeia's size, because size can actually be a really good indicator of whether or not Cassiopea is going to have more Cassiopea babies. Okay, so the larger the Cassiopea, the more higher the reproductive output. And so what we did is, in an embayment in the Bahamas, I looked insane and hauled out tons of garden fencing into this nearby embayment and lots of Cassiopea. And I set up this experiment where we had these enclosures. Still water was able to get through the garden fencing. And I manipulated slow release fertilizer to look at those nutrient concentrations. So to do that, you physically massage fertilizer in the sediment. I don't even want to know what people thought if they saw me doing this with my garden fencing and my massaging fertilizer, but that's field work. So we set this up, and we had 5 enclosures with ambient nutrient concentrations, -N here, and 5 with elevated nutrients, calculated daily nutrient loading rates for nitrogen and phosphorus. And then we added in 10 Cassiopea that we measured and meticulously identified each individual. That took a really long time. I actually tried to tag jellies using beads and monofilament, and it worked for like 4 days until the manubrium started to rip. So that was a no-go. So these were detailed notes on every single Cassiopea we had, and then went back a few weeks later to measure them again. And what we ended up finding is that there was an elevated growth rate for the Cassiopea from our nutrient-enriched plots. We saw, it's not huge, but we saw about 1/4% in growth per day in our nutrient-enriched plots. The highest growth rate that we had from an individual was close to 5% per day. And this was a little surprising. We thought it was going to be higher. What we think actually happened was the enclosures themselves, even though they were open, possibly interfered with the zooxanthellae getting energy. And so we think that that the growth rate that you see up there may actually be an underestimate of what is possible. We compared it to growth rates for subtropical and tropical jellies in the literature, and it's on par with what we're finding. But again, really interesting that you do see this increased growth in nutrient-enriched plots. All right, so overall, Cassiopea, larger, more abundant in these human-impacted sites, and they grow in the presence of elevated nutrients, more so than with ambient nutrients alone. But again, big caveat is that we are still not necessarily addressing true blooms here, right? We're not looking at polyps, which has been on my Cassiopea bucket list for a very, very long time. And then I met this incredible person who started working on this. We had many conversations about, like, if I could go back in time and one thing I could do, and she's doing it, which is super exciting. So I know she presented on this last year, but looking at how we can actually study polyps, Cassiopea polyps, and polyp development. And she and her family came up with this amazing system to actually be able to grow polyps. So huge kudos to Natalia for doing this work. I'm excited it's being done. Okay, so switching gears a little bit here and going back to Richardson's paper, one of the things that he talked about in this sort of joyride and sort of shifting, these complex food webs down to these really simplified gelatinous-based food webs, he kind of did a lot of theorizing in this paper, towards the end especially. And what he asked was, are predators keeping jellies in check? Okay, so he says it seems reasonable to presume that jellyfish are usually held in check by predation, but are they? This really cracked me up. Gathering knowledge on basic jellyfish ecology is hampered by the scarcity of scientists with an active research interest. I died. He also talked about like, and where are they going to get funding from? Which accurate. Um, so good question, Anthony Richardson. But so this really, I started thinking a lot about this, right? Okay, if Cassiopea are really abundant, what is the role that they're playing in these nearshore ecosystems? How are they interacting with other critters that are living there? Are they having this influence? Are other critters eating them? I think they are. Think it seems like that, but I don't know. So we really started getting into this, and I started talking about this in that jellyfish growth paper, which was actually published in Food Webs, funnily enough. And we started talking about, okay, I think there are things that are eating these jellies. And we do know, again, from the pelagic literature, womp, 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 that jellies are no longer being considered these trophic dead ends, right? They are an incredibly important resource for a number of different organisms, including some of the more charismatic megafauna, the sea turtles, you've got penguins eating them, albatross, bluefin tuna, swordfish, right? Lots of different invertebrates. And we've seen from some work from Sweetman, this was one of his papers he put out in 2015, that these jelly falls, when jellies bloom and then subsequently die, and they fall to the benthos, they really can drive a lot of consumptive effects, which is so interesting and has cascading impacts on nutrient dynamics and oxygen production, carbon cycling, right? So I became really interested in, well, what happens with Cassiopea and what happens if they're blooming as well? So we set out to start answering questions about the role that they play in marine food webs. And some of the work that I'm going to talk to you about is serendipitous. So this first part, where I'm going to talk about a natural history predation event that we were able to document in the Bahamas. This came about because I was tasked with being like the jellyfish lady on on a film crew. They were filming in the Bahamas for this. It's called, what's it called? Bahamas World or something. It was some sort of documentary, and they wanted to go out and they wanted to look at jellies in the Bahamas. So, okay, I guess that's me. So I took them to this site that I had been to 100 times easily to go and film Cassiopea. And what we ended up seeing is these, I'm gonna actually flip ahead, these giant venomous bristle worms. Okay, so if you know Caribbean fireworms, you know they're typically about a few centimeters long. I was telling Aki, these things were close to 1/2 meter long. Okay, they were just completely engorged. These are, I think there are 4 of them in this picture. Here's Cassiopea. So we went to this site. This is them eating. We went to this site and just saw these things, these fireworms coming out of the karstic limestone as Cassiopea were getting pushed into this mangrove tidal creek. And they were swimming like snakes, and they were whipping they have something called a caruncle, which is a sensory organ. They were whipping it around, clearly sensing Cassiopea in the water, finding them, wrapping their bodies around the Cassiopea and eating them whole. So naturally, I was psyched and thought this was the coolest thing in the world, and happened to have a film crew with me. When does that happen? So it's like, this is the best day ever. So we filmed this, and we were able to write a note on this when Frontier still had their natural history notes section. And it was just so unbelievably cool because these worms, again, typically teeny tiny, they were massive and clearly searching out these Cassiopea, an animal that we really didn't have a lot of knowledge on in terms of what was consuming them. Also really interesting because, bless you, typically these bristle worms are scavengers, so they'll find whatever they can find. Often if it's like a dead fish, that's what they'll go for. Sometimes they'll go for anemones, sometimes corals. These were clearly actively searching for Cassiopea. In fact, when I captured them, and I was telling Aki, I tried to get them with swimming after them with Ziploc bags, and they have very venomous spines. And I got one in my hand, swelled up like a balloon. That was really fun for a few days. But we started wondering if perhaps they were going after Cassiopea to try and acquire their venom specifically, if this is some form of kleptonidae. We've just started working on that in our lab, trying to figure out if we can match up Cassiopea venom with these guys. But just really unusual feeding behavior that we replicated. We went out and we used a RUV system, remote underwater video surveys, and actually put out feeding assays. So we did fish and side by side with jellyfish, and then also put out jellyfish plus fish together, and found that these worms repeatedly would go after Cassiopea only. No idea what's happening. Someone wants to work on that, add that to the Cassiopea bucket list. And so this is a little bit of that work here, right? This is a picture of that feeding assay. So we have the ballyhoo, that's dead. This is actually a live tethered Cassiopea. We went out and would film. We would randomize the treatments. We had controlled treatments as well with just the settlement tiles, and really, really, really interesting stuff. So in addition to the venomous worms that we were finding, we also found when we did these more thorough feeding assays that this adorable little fish down here, the four-eyed butterfly fish, this guy's a vicious predator, okay? So these fish would repeatedly search out for the Cassiopea. They would find the Cassiopea and they would start nibbling off the oral arms on the Cassiopea. They never went for the bell, only the oral arms. Really unusual feeding behavior. They would clearly get stung, okay? So they would go after the oral arms, get stung. All I can describe it as, they look drunk. The butterfly fish would get stung, kind of get disoriented, swim around a little bit, and then come back and start eating them again. So just absolutely unusual behavior. And in fact, in all of our videos, so several of the feeding assays where we had jellyfish only, we found that there was 100% butterfly fish presence in all of those videos, and then 67% presence in the jellyfish plus fish videos. All right. So we do see up here, this is our different treatments: control, fish plus jellyfish, and jellyfish only. We do find that there is a higher mean bite rate from the fish treatment, of course, because you have reef predators coming in to get the fish. We had sharks coming in, baby lemons. We had grouper, snapper. But again, really surprising that these butterfly fish consistently came in and nibbled on Cassiopea. All right, and this is a picture of one of them coming in hot on an oral arm. And let's see, hold on. I think I have a video. So this This one was already eaten, so the fish is already gone. So there's a NASA grouper, baby lemon shark coming in. All right. And you notice the butterfly fish only swims away just for a tiny bit. All right. So what was really interesting about this is that we looked at, okay, well, are butterfly fish going away when potential predators of these butterfly fish are coming in, right? And guess what? No, they might swim away for a fraction of a second, but they come right back in to eat the Cassiopea. So this was also really interesting.
Do you have stomach contact analysis, or are you just basing some behavioral stuff?
So this was based on behavioral stuff. Stomach is next. Stomach is next. Yeah, I know. Stomach is next. It's a great question, Mark. And at the time we didn't have permits to get stomach contents. So that's in the works. So yeah, so these guys, these butterfly fish are swimming away maybe for a tiny little bit, but then they again they're coming right back and in the presence of these potential predators. So this is the next on the bucket list of things to do. What are they getting from jellies that is worth this potential predation risk, right? What are they getting? And we don't really know. These butterfly fish do typically eat anemones and they will nibble at coral polyps. So it's not dissimilar in terms of the cnidarian prey that they're after. But yeah, we just don't really know, especially because so frequently they were clearly affected by the nematocysts. All right. So, again, to summarize, giant venomous bristleworms eating Cassiopea. We do have gut contents on those. They are packed full of Cassiopea. And for the four-eyed butterfly fish, they do seem to be going after Cassiopea, exhibiting interesting behaviors that we haven't yet documented in the literature. And of course, this is so far what we can establish, a potentially new predator of Cassiopea, not to the point that the worms are. All right. So what we're finding here is the four-eyed butterfly fish, to your point, Mark, they have tiny little mouths. They're not eating an entire Cassiopea. They're not. They are going after the oral arms. But somebody is wanting to eat them clearly. The other thing that is hot off the presses, I just got back from the Bahamas, and one of the things that we're seeing in several locations in the Bahamas is seagrass loss. We're losing seagrasses globally, but we're losing quite a bit of seagrasses in certain places in the Bahamas right now. And one of the things that has been happening is that green sea turtles that typically go after turtle grass and some other seagrasses that are here in the Bahamas or there in the Bahamas, we've noticed that there seems to be a lot of partially consumed Cassiopea in a lot of locations that have mixed seagrass beds or are losing seagrasses. We've just started doing basic surveys on this, but you can see like, oh, this is a terrible picture, but this is half of a Cassiopea gone. We saw a lot of this where you could see it being chomped. Over here, bells with some of the oral arms scattered next to the bells with oral arms missing. All right, and we saw this in fairly high frequency in some of our really turtle dense areas. And we're wondering, that paired with necropsies that a colleague of mine has done on green sea turtles in the Bahamas recently, and she's getting a lot of Cassiopea from their guts. So we're wondering if possibly there's a relationship between seagrass loss and a shift to preying on Cassiopea. So that is also on the bucket list. All right. So in sum, to just finish everything off, Cassiopea are more abundant, they're larger in human impacted areas. We have demonstrated that elevated nutrients do increase their growth. We really still need those links, though, between whether or not anthropogenic activities like elevated nutrients are increasing polyps and Cassiopea reproduction. I think it could be very cool if someone did work looking at polyps on settlement tiles and human impacted environments to see what's going on there. We know that Cassiopea are a really important part of the marine food web. Okay. This could be a potential top-down control. These critters that I've talked about could be potential top-down controls on their populations. But we do need a lot more work on how they are influenced by predators, right? How they might interact with other organisms in these nearshore marine environments. And of course, this is a call to do more work on Cassiopea ecology. All right, we know so little compared to, I would say, pelagic jellies in terms of their ecological role. And I see a lot of amazing, intelligent people that could do this work. So really call to action to try and learn more about that. Okay, and lots of people to thank. I just want to take a moment again to thank Aki and Natalia and the rest of the organizing committee for your work. You could tell it's a lot of work. So thank you. And then I have a lot of other collaborators, folks at FWC, USF, Wake Forest. I don't have my FIU team on here, but a lot of the work that I've talked about was done at Florida International University with colleagues there. And then, of course, my lab at Bentley. Thank you so much. Okay.
We have time for questions.
Do nudibranchs, are they already have?
Yes, they sure do.
Okay.
Yes, they absolutely do. So there are, it's a Dondiche species in Puerto Rico. Natalia was talking about, what was the other, you're going to talk about this in your talk, I think, the other nudibranch that you are finding near Cassiopea.
Oh, I will not be talking about it, but there's another
Dondiche
species, and there's the lettuce sea slug, Delicia, something. And then there's a piscibrine called Bursatella that Andre Morandini has found it in Brazil, and I found it in Hobos Bay, both in man-made pond, but also out in the mangroves. And if you want to know more about that too, Michaela also has a lot of info about that, and the Strader Lab has and pictures of and more information about that type of predation. But
yeah, I did not have photo documentation of this, but in the last trip to the Bahamas, there were several Cassiopea I picked up with sea lettuce nudibranchs on them as well. So something is going on. Just not sure what.
Bill Finn had the same question, by the way.
Oh, yeah. Thanks for the question, Bill. Yeah.
I have two questions. First is reasonable. So, do you see we have these giant terrifying worms? Is there actually population control there? Are there fewer? Because it looked like a pile of Cassie on the floor there.
Yes, there were a lot of them. Yes.
Yes.
So it's a really good question that I don't have a great answer to. I've studied in this system for a long time, and again, there's a lot of cycling that happens through these systems where some years you get a lot of Cassiopea, some years you don't. Some years you get a lot of these venomous bristleworms, some years you don't. So I would say in the last 10 years or so, I've seen this cycling where every other summer you get really abundant Cassiopea and then really abundant bristleworms, and then it kind of drops off. There are a couple of sites that consistently have higher Cassiopea densities and consistently have higher venomous bristleworms. There's one site in particular I'm thinking of right now. This is in the northern part of Abaco Island in a really, really contaminated seagrass bed that has some of the highest heavy metal concentrations of any place that we've sampled in the Bahamas. So I go there, but not as often now that we now know about those heavy metals. But yeah, it shifts.
Okay, and my second question is, I mean, do you think the fish are just getting drunk?
Entirely possible.
I mean, they're that's
Entirely
the
possible.
reason that- especially
I
if they
really-
do, like permanently.
It's a really good question.
We have some animals in the. We might give them a try.
I'm saying.
We saw the same behavior at Homeless Bay, we documented the same behavior from the same fish.
Yeah, it's so odd. It is, and that video didn't even do it justice. I have to go back through and get it. You see them swimming directionally towards the Cassiopea, right? They intentionally are going for the oral arms only. They get stung, their ray fins up, right? And then they kind of get disoriented and then swim back moments later. So
I'm like Dory, situation.
it hurts so good. I love that. It hurts so good. Yes.
So I know there's some debate about these, what are we calling them? The purple, bluish, orange,
Yeah.
What was the name that you?
I call them frilly appendages.
With a P, yeah.
Frilly.
With a P. Yeah, so, have you noticed whether or not the animals avoid them, are attracted to them in any way?
Good question. I have not noticed that. You mean the
Like,
fish
I'm thinking,
that
like,
hurt so
maybe
good?
because,
Sure.
like, maybe the whole debate around them being like maybe. There's a bunch of different answers for why they might exist, but like maybe it's for.
Right, you like
Like,
camouflage
camouflage
debate.
or,
Yeah,
like,
yeah.
Maybe they taste different.
I have not seen. Yeah, I have not seen that. Yeah, I haven't seen that. In the case of the venomous bristleworms, they ate everything. They
They
ate
didn't
everything.
seem that discerning.
They were not discerning. They ate it all. In some of my other work, let's see, do I have it? Oh, that's this turtle eating a jellyfish. So in some of my other work, a lot of the benthic and epibenthic critters that I've studied they're lower in densities adjacent to or right below Cassiopea, with the exception of some animals. The bubble snail, Bula occidentalis, always is abundant directly under Cassiopea. We don't fully understand why. But like seraph snails, for instance, consistently lower in areas that have high densities of Cassiopea.
Have images of like pretty much everything that was predated on.
Yes.
So like, have you sorted through like if they're?
No, also on the bucket
That would be
list.
cool, right?
Yes.
If there is this correlation.
Yes, I would love to do that.
Oh,
Yes.
do you have any ideas or evidence about stuff that might be eating other life stages, like the polyps or even the plankton?
I don't have charters. Maybe somebody else in this room could work on that. No, I don't have that. One of the big, obviously, and I talked about the big missing components here is I just am looking at Medusae. I'm not looking at any of the other life history stages. I would love to get further into polyps because I think that's where a big chunk of the story is missing. But especially for who is eating them as well. Yeah, Mark. Oh, sorry.
No, I was gonna say, you don't think they're eating them, right? Do you? Like.
Collectively.
Yeah, female. Yeah.
Okay, so I just have a like, quick comment.
Yeah.
That in our limited experience.
Yep.
At least in the Florida Keys. Forget about predation. One big nor'easter, one big.
Wipes them out.
Weather event. and
Yep.
They all get wiped out. And
Yep.
they go somewhere.
Yep.
An ocean, whatever.
Yep.
And then you have a population bottleneck and then the polyps have to, so it strikes me, if you're interested in population dynamics. These gross weather patterns and maybe climate change, I don't know if we would invoke that, but these are more likely to impact Cassiopea population.
Yes.
Any multiple.
Totally. I just think critters are cool and critters eating other critters is really cool. But yes, absolutely. And some of the work that we're interested in doing, and I've talked to a few of you about it here, we're seeing that with ocean warming, especially in a lot of places that I work in the Bahamas and South Florida, we're actually seeing that at high enough temperatures, Cassiopea are melting. So I was struck this past trip to the Bahamas. I was just there a week ago. And in several of my field sites, you walk out in the water, it's brown goo. Yeah, yeah.
They're beautiful, but they're dead.
Yes, yes, yeah, and I am with you. And where I've spent a lot of my time working in the Bahamas, particularly in the northern Bahamas, they were hit really badly by Hurricane Dorian in 2019. It was a Cat 5 hurricane. Cassiopea got wiped out. Yeah, they're back now. They're back and really, really dense in all of the usual haunts, the places that we see them.
It would be interesting to population.
I would love that.
Big massive,
Yes, I would love that.
that's a bottom there.
We have a.
Oh, sure.
And then.
Sorry.
Yes,
Sorry.
yes.
This is another question from Bill. So he says butterfly, fish, fireworms, turtles are usually found out on reefs. Could their predators be responsible for the mangrove seagrass? Sorry, could these predators be responsible for the mangrove seagrass distribution? That's good.
Could these predators be responsible for their distribution? I think to some extent, maybe, but I'm with Mark here. I think that the bigger drivers are probably going to be more abiotic than biotic. Where we see a lot, frankly, where we see a lot of Cassiopea, it's like I said, the usual haunts. These are, silty, sandy, sediment dominated sites, relatively low wave action, typically higher nutrient loads. Temperatures kind of have to be just right, too hot, and you won't see them. I do think that some of the distribution is a function of predators, but I don't have enough information under my belt to say that definitively. And a lot of the sites where we're doing this, these are in mangrove tidal creek systems, so they're not out on open reefs. These are protected systems that are all tidally influenced. Yes, yes.
So you said that you had experienced Cassiopea melting in the Bahamas?
Yes.
What temperature was that happening at?
Ooh, it was like 29 degrees Celsius. It's
29 Celsius.
yeah, yeah,
Was that, did you find that at multiple sites or just one?
multiple,
Okay, and was that consistently within this like 1 year or across a couple of years?
several years.
Interesting.
Yeah. Yep. Yeah, I've been talking about Cassiopea melting for a very long time. One of my sites, in fact, and I didn't talk about this project today, but I was doing a Cassiopea removal project in some large seagrass beds in the Bahamas. And so I was trying to better understand benthic community dynamics and how Cassiopea would influence them. This was a site that had I can show you this site, actually. Hold on. Where's this picture? This is the site that within basically a meter squared, you see Cassiopea on top of Cassiopea, right? I don't even want to estimate how many are in this particular image, but a lot. And so in this site, we went in to do a removal, like just scoped it out one day, came in the next day to start actually doing the work and taking the Cassiopea out. And it had been really, really hot the previous day and into the next, and all of these were melted. It was goo. So I had to scrap that part of that project. Yes. Nope. These are all really, what? Have I done any sort of like analysis on the melted tissue? I have not.
You can do that in a lab.
Yeah, but the lab is less fun, Mark. The field is where it's at. Sealed is where, yes, I have not yet done that.
The thermal adjusted,
But-
instrument.
Yeah,
Any more questions?
yes, any other, oh, yeah.
In the fertilizer experiment.
Yeah.
Higher nitrogen, did you see any changes in their color, like their algae were getting more dense or anything?
We did not, in that particular experiment. I have done zooxanthellae work where I've enumerated zooxanthellae densities from Cassiopea in nutrient enriched sites, and they are more abundant, and the coloration does seem to be darker. Really beautiful, like dark purple, almost black. And yeah, but for that project, we didn't look at coloration.
Victoria.
Yeah.
Do you have an idea in the dense human areas or densely populated human areas, if the Cassiopea are there because the humans chased out other species first and then they kind of moved in? Or if the Cassiopea being there might have caused other species to then make them here?
Yeah, Victoria, that's such a great question. It really is like chicken or the egg, right? What's happening in these sites? Is it sort of our critters going away because they're impacted sites to begin with? We tried to address that in that first project, all the way back. That I talked about where we were looking at grazing intensity. So we experimentally manipulated Cassiopea densities and nutrients, essentially replicating disturbance, and established baseline benthic and epibenthic communities. So saw what was in the system, right? And did that over a period of a couple of years, and then came back in, oh, that's not the right one, came back in and then experimentally manipulated Cassiopea by physically adding thousands of Cassiopea into this system along with nutrients, and critters moved out. So we've tried to address that, but it is often something that I grapple with, right? Are what we seeing sort of just a byproduct of the collective impact from humans, from anthropogenic activities, right? From dredging, from nutrient loading, from all of these things that we're doing. Is that the real impact here? I tried to get at it, but yeah,
I mean, who are
great.
they competing with in a soft sediment?
Right, right.
Hard sediment is like everybody wants to.
Right. We have found that in a lot of my work is they are competing with seagrasses. So there's that pretty critical organism. And they are resting a lot on seagrasses, especially when they're really abundant. They are on top of seagrasses. We also very recently, and I don't want to speak too soon, but we're writing a paper right now. We found Halophila stipulaceae, the invasive seagrass in the Bahamas, and we're finding Cassiopea are very abundant in and around and on top of the Halophila. So that will potentially have interesting interactive effects, I would imagine, as Halophila becomes more prevalent in the Bahamas. So yes, there's something going on there. Yeah.
So thank you, Betsy, one more time.
Oh, thank you.
Wow.
Thank you.
Well, now we have a break, so if you have more burning questions, comments for Betsy, go ahead and do that, and then we'll be back at 10.
Yeah, so we'll start our next next session at 10:30, so be up before that so we can get started right away. We'll try to get some coffee and things going downstairs and have some of our leftover snacks available as well. So we'll be back at 10:25.
Aki, do you want me to just sign out of Zoom, or do you want to stay on Zoom? What do you want to do?
out.
Okay. All right. I didn't see if there was anyone else in the, oh, that's Bill.